postdoc position as a vet postdoc. So she has her veterinary degree and she's finishing up her postdoc at University of Florida. And she's going to tell us all about her research and how she sort of married her career veterinary work with her research. So thank you.
Cool. Hi, everybody. Thank you for coming. Everybody's in the back over there. Like Ruth said, I am a veterinarian. I specialize in aquatic animal medicine, and I'm also a researcher. I like to point out that I've spent more time doing graduate work in molecular field than veterinary work. A lot of people just pin me as a veterinarian, but I am a biologist and a researcher, and my talk is kind of split up into those two different things. So I'm going to go into a pretty in-depth background about myself, so you guys all know where I came from, and then we'll talk about my research a little bit. So what I'm going to talk to you guys today about is what sea lions can teach us about herpes viruses, cancer, and environmental health. And as a veterinarian, I like to think that I'm not just an animal doctor, but I utilize the animals and wildlife to better understand what's going on in an ecological scale. So I'm going to be talking to you about sea lions for the most part today, because primarily over the past 3 years, that's the major species that I've been working with. But this can be translated to dolphins, it can be translated to fish, it can be translated to mussels and inverts. So although we're talking about sea lions right now, a lot of the techniques that I'm talking about can be applied across the board. So we will just get to it, if I can advance. Let's see. Is this on? Sorry. That's okay. That's on. Oh, I felt that. Do I point it? Yeah. There we go. Which button was that? Just that one. Okay. All right, so like I said, a little overview. I'm going to talk about a background of myself, and then I'm going to talk about my clinical experience and kind of where I came from and where I learned what I know right now. And then I'll talk about my PhD experience, and that's what I'm doing right now and what I'm continuing to learn. So my background is I grew up in South Florida area, kind of on the water in West Palm Beach, and I did my first 2 years of undergrad at Auburn, actually, so War Eagle. Sorry, I know not a lot of you are Auburn people, but some of you are. And then I actually moved back down to South Florida to complete my bachelor's. I changed my major from pre-vet to molecular biology, primarily because I didn't feel like I was hitting the grades to get into vet school, unfortunately, in my general chemistry and organic chemistry classes, so I have a high respect for those of you that do chemistry. It was a struggle for me. But I do love molecular biology, and that's something that's kind of held true throughout my career. After I finished my degree in molecular biology, I was kind of undecided about what direction I wanted to go. I loved cell biology and genetics, but I also really loved marine animals and aquatic stuff. So I was trying to decide if I wanted to go to vet school, if I wanted to be a geneticist, or if I wanted to be a marine biologist. So I took a little time off between my bachelor's and my master's, and I was a biologist. I worked in a genetics lab where we did primarily Drosophila genetics, looking at cancer cell signaling stuff in fruit flies. Very glamorous. And I also worked for West Palm Beach Environmental Resource Management, and there I did primarily sea turtle stuff. We were doing sea turtle nest monitoring, beach renourishment, effects on the nests within the sea turtles, as well as the effects of hurricane accretion and erosion of different beaches in the southeast part of Florida. I also volunteered and then was hired on at Harbor Branch Oceanographic Institute, and I was a part of the Marine Mammal Stranding Network down there. So we did a lot of cetacean and whale stranding and necropsies. During my time there, we never actually rehabbed an animal, but they do now. And we also did the dolphin health assessments. So before I went to vet school, I was a vet tech for dolphin health assessments, and then after, oh, you can't see my turtle in that picture. Somewhere in that picture, there's me holding a turtle in the water on the right. But during my experience at the West Palm Beach Environmental Resource Group, working with sea turtles, I got exposed to sea turtles with fibropapilloma. And that's a herpes-induced cancer in sea turtles. Usually happens in juvenile turtles, and it's associated with immune suppression. And if you guys have seen that or know what it looks like, it's quite striking. The tumors are really significant, and when I first saw it, I couldn't believe that this was something that was happening right in my backyard and I didn't know about it. So after doing a bunch of necropsies on those animals, I decided that I was going to pursue my master's in trying to figure out what's going on with that disease and get a better understanding of how the herpes virus is inducing that disease and how the immune system is tied into that. And that's kind of going to be a common theme throughout my talk. I have probably an unhealthy obsession with herpes viruses. I think they're the coolest viruses in the world, and hopefully by the end of this talk you guys will at least think they're all right. Maybe not as cool as I think they are, but they really are cool. So I got my master's at Florida Atlantic University, which is where I got my bachelor's too. I had a really good network of people there, and they do a lot of really good work there, so I stayed there for my master's. And then I went to vet school. I decided that a lot of the sea turtles I was looking at throughout my master's degree were very anemic, and I didn't understand why. So the higher the tumor burden on the animals, the more anemic they were, and that was something that I noticed just haphazardly throughout the processing of all of my samples, but I didn't really understand why. Now I know it's because they had a chronic disease, and that's something that happens with chronic disease, but I wanted to be able to figure out how I could better understand systems as a whole before I specialized into one little area, specifically viruses and cancer-inducing viruses. So University of Florida has a great aquatic animal health program. It's one of the few universities that actually has a certificate. It's actually, I think, the only one that's associated with aquatic animal health. So throughout my 4 years there, I got to do manatee health assessments, dolphin health assessments, sea turtle cold stunning rehab and response, a bunch of fish work with the Tropical Aquaculture Lab, and there's a couple really big ornamental fisheries and aquaculture farms in Florida, so I kind of got to jump around and get a taste of aquatic animal medicine throughout my DVM. And then I got pushed kind of by all of my mentors after I finished vet school into doing a small animal internship. I had never worked with cats and dogs in my life, except, you know, when I was in vet school, obviously. But I was very intimidated by cats and dogs, and owners particularly. But it was the best advice I could have ever gotten. Working with small animals, you have the ability to really practice a high level of medicine. You have access to the animals, and you can do things in domesticated animals a little bit easier than you can in wildlife or aquatic animals. So just getting my medicine down during that year was really, really important for me, and my internship was mostly emergency medicine and surgery. And I specifically picked that internship because it focused on those things, because in aquatic animal medicine, that's a lot of what you're doing. So following my small animal internship, I spent a year doing a specialty internship. That was with the National Marine Mammal Foundation. Some of you may know of them because they come over here and do dolphin health assessments in Barataria Bay, and kind of hang out in this area sometimes to check out what's going on with some of the animals in the oil spill. Some of the work that you guys do is associated with them. But it was split up between the Navy Marine Mammal Program and SeaWorld. And I'm going to talk a little bit about my experience at those facilities. For those of you that don't know, our armed forces actually have soldiers that are sea lions and dolphins. The main base is in San Diego, and that's where I worked. And those sea lions and dolphins are responsible for doing diver defense and finding underwater mines, as well as doing protection of some of our ports. So my job as a clinician there was to help take care of the health of those animals. And it's one of the best places to learn aquatic animal medicine, in my opinion, because they have unlimited resources. Those animals are considered soldiers, and they have the same health insurance as anyone that is enlisted has. So we had full access to the Balboa hospital that was there, the human hospital, so we could CT dolphins whenever we wanted. We could go raid their closet for supplies whenever we wanted, and we got to use their fancy machinery, their ultrasounds and stuff pretty often. So it was a great place to learn how to practice aquatic animal medicine. I will say the caveat is a lot of aquatic animal medicine is not done to that level. So although I do know how to CT a dolphin, you don't necessarily always get to CT your patients. So it's good to be able to know how to do those skills and utilize those things, but practically speaking, most places aren't going to have those resources. A lot of it was preventative medicine though, and the trainers are absolutely incredible, and the animals, because of that, are so well behaved. So I got a lot of experience doing ultrasounds, which is a really non-invasive, very important way to understand what's going on with the health of an animal alive. And it's really important in this area right now because the dolphins are experiencing a lot of pulmonary lesions secondary from the oil spill, a lot of adrenal gland lesions, a lot of reproductive issues. So during the health assessments, the ultrasound is one of the most important things that you can do as a clinician to get a better understanding of what's going on inside of that animal. And then endoscopy and anesthesia and surgery. Surgery is something that, or anesthesia I should say, is something that isn't very common in cetaceans or dolphins. And I was lucky enough to be a part of 3 different anesthesias. And now it's something that they're starting to do a little bit more regularly because they're figuring out ways that they can safely anesthetize animals. But it was really cool to kind of see that process of figuring out when we're going to decide to take the risk of doing that anesthesia and how that all played out. It involves a lot of specialists. Most of the vets kind of stand back and hold up the walls while people that know more about us step in and do their thing. But it's really cool to see the teams come together. And then these animals are soldiers. They do get deployed to go on trips to help find mines and stuff. So we did a lot of animal transport, which has been very useful for me now during my stranding response periods. So SeaWorld was also an incredible opportunity to work. They have a huge variety of animals right at your fingertips. They also have a really high caseload. So it's kind of like a small animal internship, but with aquatics, because you see a ton of animals and you learn a lot and you have a lot of resources. SeaWorld dumps a bunch of money into making sure that their animals are well cared for. So we were able to call in specialists when we needed to do things like eye surgery in a dolphin, and we always had really nice equipment and ability to treat animals to the level that was necessary in order to maintain their health. SeaWorld is not just marine mammals. We actually did a ton of fish medicine. I would say about 50% of my time was spent doing fish medicine, and I love fish medicine. It's kind of actually where I thought I was going to end up and not in the marine mammal world. So they have a huge collection of elasmobranchs, and my favorite animals are actually rays. So I kind of drifted towards doing a lot of the ray medicine, developed a really good preventative health care for them, and we started doing some reproductive stuff with them. We got them trained to go into stretchers for ultrasounds, got them trained to take medications. Rays are really smart. People don't give them enough credit. And then we also had larger elasmobranchs. The bottom left picture is a sand tiger that had some skin issues that we take biopsies from, and then we have a ton of other animals, eels, cephalopods, and then inverts as well. In addition to that, we have a bunch of sea turtles out on the west coast in the facility. There was a couple accidental clutches at SeaWorld that got laid in this tiny little sand of a beach, and we weren't allowed to release those hatchlings, so we actually ended up with about 100 sea turtles. And that causes some issues, especially as they all ended up being male because their sex is temperature dependent and it was a little cold out there. So they all turned into boys, and once their testosterone started kicking in, they tried to show who was the biggest turtle in the tank, and they would kind of hurt each other occasionally in doing so. So it was a lot of reptile medicine too and trying to maintain that. And they have a really good program for breeding dart frogs and a lot of endangered species, so I got to do a bunch of amphibian stuff too. So a lot of that. And then birds. It's something that SeaWorld should actually be considered more of a bird facility than a marine mammal or aquatic facility. They have more birds than fish or marine mammals combined. A lot of which are penguins, but they also have beautiful collections of a ton of different species. The aviculturists are absolutely incredible, and I've learned so much from them, as well as the aquaculture, or the aquarists at SeaWorld. They're super knowledgeable. They really taught me a lot about the husbandry of it, and 99% of your problems in captive situations can often be at least improved by husbandry care for the animals. So it was really important to tap into those resources, and I am forever grateful. And then lastly, at SeaWorld, they do a lot of rescue and rehab. It's something that people don't realize, I think, how much they do. I would say about 40% of my time was spent dealing, and I was there during an unusual mortality event of sea lions, so I should preface that, but we would go out and dart sea lions that had fishing line entanglements. We would rescue any cetacean or whale that showed up on the beach. And the rehab and the rescue part of it was something that I got really excited about. I had previously seen the cancer in sea lions that I'll be talking about today at a place called the Marine Mammal Center, which is where I currently work. But at SeaWorld was the first time that I had to be the doctor on some of these cancer cases, and it was something that was really hard on me because a lot of them come through this SeaWorld for rehab, and there's nothing we can do about it. They strand in advanced stages of disease, so you end up having to euthanize them. And although we did take samples on those animals and we did do kind of basic research and assessment of them, it was a little disturbing to me that we couldn't do more. And I felt like we had access to these samples and we should do something with them. So that's when I decided to do a PhD. So I've been in school for a really long time, but it's been really great because it's enabled me to do the things that I want to do. The Marine Mammal Center is a facility that's in Sausalito, California, so that's just north of San Francisco. They have a stranding response zone of about 700 miles along the California coast. There's a ton of marine mammals out there. They are the largest marine mammal rehabilitation facility in the world, and that's because they deal with a lot of sea lions and seals. So the Marine Mammal Center has been seeing a high incidence of urogenital carcinoma in California sea lions since they began. And so Dr. Francis Gullen, who is my PI that's over there, had kind of brought me into them because of my background with herpes and molecular stuff and my interest in marine animals, and offered me basically a full ride. Fully funded PhD, would fund my research, and they basically had a project pretty laid out for me. And for those of you that are PhD students or past PhD students, and now you have successfully survived the trip, you'll know that the funding is half the battle. So I really started off on a great opportunity, and that's kind of something that's hard to turn down. So part of my reservation about doing a PhD is I just worked really hard to learn how to be a vet, and then I was going to bury myself in a lab doing molecular stuff with pipettes, and I did not want to forget how to be a vet. So they really catered to me and let me kind of mold my PhD into something that allowed me to continue to practice medicine, and I'll talk about that a little bit too. With that fully funded PhD, they told me I could take it wherever I wanted. And like I said, I had been in school for a really long time. So I went to that school at University of Florida. I've established great relationships with people at the University of Florida, and I knew the lab that I worked in there. And I knew that that would expedite me being able to finish my work as quickly as possible. As well as the University of Florida has a great cancer genetics institute associated with their medical school. So it just made sense to me, although geographically it doesn't, because I am bi-coastal. I spend about a third of my year out in California and about 2/3 of it, maybe a little bit less, in Florida doing research. It was the best place for me to be to do what I wanted to do efficiently and effectively with the resources that they had available. So that's how I ended up kind of being in both places. It's a bit confusing for everybody, including myself. So like I said, my clinical work was really important to me during my PhD, although it did slow down my research a little bit because when I was a clinician, I was a clinician. And unfortunately, during my past 3 years, there's been an unusual mortality event of the sea lions out in California. So not only was I responsible for helping them during their busy season, but it was their busiest busy season that they've ever had for the past 30 years that they've been open. They usually see about 300 to maybe on a very busy year 500 animals, and over the past 3 to 5 years they've been seeing about 1,600 to 2,000. So they unfortunately did not hire any more help for that though, so that was the staff veterinarian, Dr. Carr Fields, myself, and the intern that would come through to help take care of like 5 times more animals than they would typically see in a year. So when I was there, I was very busy. The majority of those animals that came through were malnourished pups, so yearlings, 1 year and younger. And that's because there's been some warm water events. I don't know if you guys know about this thing called climate change, but the water out in the California coast has been unusually warm. And then on top of that, there was also an El Nino. So within that time, as a result of that, the prey fish, we think, is what we're postulating is happening, is the prey fish have moved to areas where the water is more comfortable for them. Unfortunately, the mama sea lions can't get out to them and then back to nurse their pups in an appropriate time. They also can't get enough calories to produce the quality of milk or the quantity of milk that's necessary in order to wean their pups to a healthy age. Usually they wean them at about 9 months, but we noticed that they were coming in January instead of in March. All sea lions are born in July. So it's nice. They sync their births for us, so it's very easy to tell how old they are, for the most part. So all of these malnourished pups were showing up on our beaches because the moms were either abandoning them too young, or they weren't producing good enough quality milk for them to put on the weight that they needed to in order to start foraging by themselves and successfully wean. So on the right, you'll see that's a necropsy picture of a sea lion. If you haven't seen a sea lion necropsy before, it probably doesn't look that weird to you, but that liver is extremely pale. It's hepatic lipidosis. It's what happens when your body is starting to use up all of your fat sources, and that animal has no subcutaneous fat. Although sea lions do not have blubber, they do have a very thick layer of subcutaneous fat, and usually it's about 3 to 10 centimeters, depending on the age of the animal, and that animal has 0. So that's pretty much what all the necropsies showed on all of the pups, but that definitely kept me very busy in the springtime throughout my PhD. In addition to that, when water gets warmer and when there's increased rain events, nutrients go into the water, and algae love that. So if it's a warm water environment and there's additional nutrients, there can be large algal blooms. And on the west coast, we have something called domoic acid, which is pseudonychia, a toxin produced by pseudonychia in algae. And that is what causes paralytic shellfish poisoning, which I'm sure a lot of you are very familiar with, in people. In sea lions, it causes pretty much the exact same thing. So the sea lions, that sea lion on the left is in apostotnik, or is in an apostotnik position, head back, flippers together. He's actually actively seizing. So we get a lot of adult and subadult sea lions that come through during these warm water periods when there's these algal blooms of seizing sea lions. So on the right is a cross-section of a brain at the level of the hippocampus of a sea lion, and you can see the left side, am I getting my lefts and rights correct? Yeah. On the left side is a normal hippocampus, and on the right is that same animal, but it's an atrophied hippocampus. So the hippocampus is what's responsible for your ability to understand where you are in space, and it helps with memory. So now that there's been the first domoic acid bloom that really affected the sea lions was in '98, and it seems like as the blooms are coming, they're becoming more frequent and they're becoming more toxic. So the animals are seizing more and they're having more chronic exposures, and that's actually resulting in, instead of this animal on the left is in really good body condition and having seizures, we can pull them out of that. We give them a bunch of fluids, we give them some anti-seizure medications, and after about a week they're back to normal. We can release them. Now that they're being chronically exposed, it's becoming a much bigger issue. It causes issues with their hearts, and because their hippocampus is basically atrophying away, they don't remember where to find fish anymore, we're postulating. So now these animals are coming in poorer body conditions because they're not able to hunt, there's less fish available to them, and on top of that they're having cardiac issues, so they can't swim as fast to catch the fish that are around. So the reason why I'm bringing these two specific things up, these are two of the main reasons why we see sea lions strand, but this also goes to show how sea lions, marine mammals, aquatic animals in general can really act as sentinel species for an aquatic environment and really get a better understanding of what's going on an ecological scale. Because although these sea lions are cute and adorable and it's really easy to get the public interested in what's going on with them, we're totally just using them to get people interested in what's going on in a bigger picture of the environment. So I'm going to talk a little bit about my research too, which is a way that we're doing it, but my research is less directed about environmental impacts. The malnutrition in the baby sea lions is, a poster child for what we're going to have to worry about when it comes to global warming, as is these domoic acid outbreaks and blooms, and that's a huge effect on the fisheries out there right now. They close fisheries as a result of this pretty regularly, and it's becoming more often and longer term. So I also got to do a lot of really cool field work. I spend about 1 to 2 months out in the field, and that is literally out in the field. They bring us to an island in the middle of the Pacific and they drop us off for 2 weeks to a month, and then they come back and pick us up. On the left, that's our portable anesthesia kit. That's a little pelican box, and we can anesthetize probably up to about 120 kilogram sea lion with that. And so I've done field work for a variety of reasons. Basically, they farm us out as contractors for biologists that need a vet to basically make animals go to sleep so they can get the samples that they need. So that's my primary role, as well as doing a health assessment on those animals to see their health status so they can interpret their data appropriately. But we do a lot of stable isotope work. The animal on the right is actually a Guadalupe fur seal, very highly endangered pinniped. I think perhaps the most endangered pinniped. And they've never been tagged before. So I spent a month on Guadalupe Island in Mexico, which is in the Pacific, not the Guadalupe Island in the Caribbean, putting tags for or actually I just was putting the animals to sleep and the biologist was putting tags on them. But very expensive satellite tags, as well as some GPS tags that are like $5,000 a piece. I had no idea they were so expensive. But those animals now, we officially know, I think we tagged 38 animals and we processed 100. So we have an understanding of how their health is being affected by this warm water event, in addition to understanding once they're weaned, and we did everything from pups all the way up to subadults. These guys get really big, so once they get past the subadult stage, they're just not handleable unless we're darting them, and we didn't want to take that risk with these animals the first time we were out there. So we stopped at subadults. But now we're kind of unlocking the mystery to where these animals are actually going to forage. And it was the Oil Wildlife Care Network actually that funded this project. It was like about a $200,000 project, because they're trying to figure out what areas they need to protect forage ground-wise in order to protect this species a little bit better. And it come to find out they're leaving Mexico waters and going all the way up to like California, Washington area to do a lot of their foraging, and that was something we really didn't know before we did this work. So I really love field work. We also do leptospirosis monitoring in some of the animals, which is a bacteria that is zoonotic that people can get that causes renal failure. And we do survivalship studies. So we'll anesthetize the animals, we'll brand and tag them, and then the biologists go out and do resightings on those animals to see how each year is doing health-wise and survivorship-wise. Stranding and necropsy, probably my favorite things. I am obviously very active in the stranding network in California, with mostly sea lions and seals, but we do get a ton of large whales. On the picture on the right, that's the Golden Gate Bridge in the background, and that's a humpback whale that came in on the front of a large boat, unfortunately. And then the Coast Guard dragged it to Angel Island, which is where we ended up necropsying it. I would say we probably do about 5 to 8 large whale necropsies every year, and that's my favorite thing to do, to get into a large whale and necropsy it. So in Florida, we just got a NIFWIF grant about a year ago to set up a stranding network in Levy, Taylor, and Dixie County, which is the west coast of Florida, kind of northwest coast of Florida. It's a very unique and challenging environment. It's a lot of marsh. It actually kind of looks a lot out here, except for we get major tidal fluxes out there. So we've had a handful of disentanglements that we've done. We've had a manatee, and then we've had a mass stranding of pilot whales. 10 pilot whales showed up on the beach. So it's a very new system, and we're just getting everything set up and going, but that's the fun part. I'm sure I don't have to tell you what these awesome facilities you have, but you get to, pick what you want and get new toys and stuff, and it's been an interesting process to see that all work out. It's also been really great collaborating with the locals. Like I said, the waterways there are super dangerous, and us showing up from Gainesville, Florida, have no idea where the oyster beds are and where they're not. So being friends with the locals and having them actually take us out on their boats when we need to go respond to animals has been the key to our success, particularly with that mass stranding of those pilot whales. In that mass stranding, unfortunately, there was 10 animals. We euthanized 2 of them. One of them we did rescue, transport, SeaWorld, rehab them, and we released that animal. It's the first pilot whale in a real long time that has been successfully rehabbed and released. Her name is Gail. She's hanging out, or at least the last time her satellite tag fed, off the coast of North Carolina. So we released her in the Gulf, and she went all the way. It looked like she was going to go straight into Cuba. Right before she hit Cuba, she took a hard left and then went all the way up the East Coast to North Carolina. So that was a really cool part to see. And then on the other side of it, the other 7 animals were found dead. So we necropsied all of those animals and tried to figure out what was going on with them. But I really do enjoy stranding response, and I love necropsies because I feel like that's where all the answers really are. So I did have to do some research for my PhD. And like I said, that was in something called urogenital carcinoma in California sea lions. So we wanted to know if basically you could use them as a comparative model for studying virally induced diseases. And there's this new kick with the NIH using something called true disease models. And basically, it's really nice to be able to study the disease that you're interested in the species that gets that disease. But obviously, with human diseases, there's limitations to what you can do study-wise. And so a lot of research happens in the lab with lab rats and monkeys and things of that nature. When you're doing research like that, especially with cancer research, a lot of times you have to suppress those animals' immune systems. And unfortunately, cancer, when it develops in an environment, is really affected by the immune system. So they're already starting out in kind of a model that isn't representative of how cancer is really developing in a human or in a real, true species. So although there's a lot of confounding variables studying cancer in sea lions, because we can't control every parameter that's associated with them, it more represents what's happening in the real world. So I'll talk about it in much more detail, but the idea of having an animal that has an intact immune system that also has a variety of cofactors that are feeding into something that is truly a multifactorial disease is really important in getting a better understanding of what's happening in the system as a whole and why it's happening. So urogenital carcinoma, it's not just a viral infection. A lot of sea lions have been screened and have tested positive for this viral infection, but they don't necessarily develop cancer. So it's actually the purpose of my PhD was to try and determine whether or not this herpes virus was actually associated with the development of the cancer or the transition of the cells to a neoplastic state, or if the herpes virus was just kind of along for the ride. There's been a lot of research in other areas trying to understand why these cancers are happening, and contaminants is playing a major role in this. DDT and PCBs within the Southern California bight are really, really high. So they've done some research and found that the sea lions that have cancer have an 85% higher level of PCBs in their system versus animals that don't have cancer that are within that same cohort. And then there's been some research, and they're still trying to work it out, that there might be a genetic predisposition, just the way that sea lion breeding territories are set up. There's one guy who's kind of a big deal that all the ladies want to be with. And so there's a lot of inbreeding that actually happens within the sea lion population, and they've associated this specific MHC class 2 molecule, which is an immune cell receptor, that is associated with potential predisposition to developing this cancer. And then there's hormone influences. And for those of you that have done work with PCBs, you know that they can be estrogen mimics. So the fact that these animals have something that kind of acts like an estrogen within their system and they're getting a urogenital cancer, the question was, are these estrogen mimics helping to proliferate this cancer or driving the development of this cancer? So Dr. Katie Colgrove, who's a pathologist, a marine mammal pathologist, got her PhD kind of trying to tease out understanding the impacts of hormones and other oncogenic receptors within the cancer. But my focus is on the virus, because the herpes virus is the coolest part. So for those of you that have not seen reproductive tracts of sea lions, on the left is a lady, on the right is a boy. And urogenital carcinoma, the primary tumor, starts within the genital tracts. So it starts within the cervix of the females or the penis of the males. It can be used as a model for cervical cancer in people. That's caused by a papillomavirus. That is not a herpes virus, but they actually exploit the same cell pathways that actually induce the cellular transformation that predisposes the animal to developing cancer. So although it's not the same viruses that's inducing the cell proliferation, it is the same mechanisms behind it, if that makes sense. And something that it's probably even worse on this screen, but that I wanted to point out is these lesions, these primary lesions are not very impressive most of the time. If you were to look, that's a cervix, so the bottom part of the picture on the left is the vaginal canal, and then that little bulb is the cervix, and it's kind of filleted and like laid open. That's actually a pretty significant lesion. Can you guys see my pointer right here? And that's hard to pick out. So if you don't know what you're looking for, you may miss that. And the penile lesion is just this roughened plaque right here. So both of these animals had extremely metastatic disease. So just with those tiny little primary lesions, that cancer has spread throughout their body. And it's really the metastasis that kills you in cancer. If you have a primary lesion, it's really not that big of a deal. You can get it cut off surgically, and then maybe a little bit of chemo, and then you're good. If it hasn't spread throughout your body, chances are you'll be fine for the rest of your life. It'll never come back. It's when those cancer cells develop the ability to pick up and go to other places in the body where it causes a big issue, where it's more systemic, and that's a much harder thing to pull people out of. And I will say, and this is my little soapbox just across the board for studying cancer, lab animals are not the way to study metastatic disease. The NIH actually doesn't fund a lot of metastatic research because it's not translatable within traditional lab models. And that's because that microenvironment isn't there. The immune system, the cancer is basically battling against the immune system to learn how to hide from it in order to start spreading to distant places. And without having that intact immune system, you're not creating the same environment that would be happening in a natural setting. And that's really where I think studying naturally occurring true disease models, that's where the key is. 90% of people with cancer die of the metastasis of the cancer. So if you can just figure out a way to stop the spread of that cancer, then you really can develop treatments directed towards ending mortality, as opposed to just figuring out how to stop breast cancer or liver cancer. It's really ending the metastasis that is probably going to be the major breakthrough in the future in order to help cure cancers. And a lot of that is probably going to be associated with controlling the immune system. So on the left, that is a urogenital tract of a sea lion. So the kidneys are the two round things on the top. The ovary, this is what healthy looks like. This is what disease looks like. This animal was nice enough to just get unilateral disease, so it makes it a really pretty picture. So this is what a normal kidney should look like, a normal ovary. This is the uterus right here, and then the bladder is right here, and this is the exit. And then on this side, this is a metastases. So the primary tumor is down here in the cervix. It's spread, and this is an enlarged lymph node that's actually wrapping around the ureter of the kidney, so that the ureter is the tube that drains the urine from the kidney into the bladder. And this is one of the ways we pick up this disease clinically in animals pre-mortem, is hydronephrosis, that backup of urine into the system. So it's this that kills the animals. It's not that primary tumor that's in the cervix. And it always amazes me, I probably don't have to tell you because you guys all work with animals, but how tough these animals are. When we open sea lions, they're pretty much just a swimming tumor most of the time. And the fact that they're able to not only live with that long to get to that point, but some of them are even reproductively fecundant, where they're lactating, or they have a placental scar that tells you that they've reproduced within that year. That's something that's pretty incredible, but I also think that's something that points to the fact that these animals, the metastases happen very quickly. There's something that's driving this tumor from spreading, and that spread and the spread of the disease happens in a pretty fast manner. And then on the right is just a liver riddled with tumor. It's really congested and hemorrhagic, and all of the white circular plaques you see are metastases. So that liver is pretty much non-functional at this point. And then this just goes to show you the sea lion's head is here. He's kind of laying to the side. So head's up here, tail's down here. This is the liver right here. Kidney. This is a mass, a metastatic mass that's taking up the entire abdomen of that animal. And you would be surprised. As a clinician, it actually makes me feel really bad about myself when I ultrasound these animals, and I can't see that on ultrasound. I don't understand how I can't pick that mass out, but you can't. There's a bunch of guts in the way, and when you open them up, you're just like, how did I miss that? But I don't like euthanizing things unnecessarily, so I really want to know that that animal has an advanced stage disease that I can't fix before I decide to euthanize it. And this disease is something that's really, really hard to say that it has. So unless they have that hydronephrosis, I can't say for sure that that animal has cancer. So I just kind of wanted to point out how extensive this disease can be, and pre-mortem it can be really difficult to pick up. And I will also mention that urogenital carcinoma is not the only cancer that sea lions get. Here is a list. We did a retrospective paper looking at the cancers in the past 10 years that have presented to the Marine Mammal Center. And we kind of see it, mostly reproductive cancers like leiomyomas. Those are benign tumors of the smooth muscle in the uterus most of the time, in the sea lions at least. And then we see a handful of lymphomas, and then some spindle cell and some adrenal carcinomas. But none of these are very significant, and only the lymphomas were responsible for causing death in these animals. The rest were just incidental findings. But I will say, of all the animals that we looked at over the past 10 years, 90% of them had urogenital carcinoma. So it is way more represented within the cancer that we're seeing in these animals than any of the other types of cancers that we see. So this list kind of represents what you would expect to see as noise in cancer associated with any wildlife species. Like, you're going to get a little cancer here and there if you live long enough. But the urogenital carcinoma is not that. We see urogenital carcinoma in much younger animals, and it's represented at a much larger scale within the population that strands. So here's just a little breakdown so you guys understand the prevalence of it. There was a paper by my mentor, Dr. Golan, that looked at urogenital carcinoma from 1979 to 1994, and that showed 18% of the subadult and adult sea lions had urogenital carcinoma within that timeline. I will say that all of this research excludes pups and yearlings, and that's basically just because they're not sexually active yet. This is thought to be a sexually active transmitted virus for the most part. And usually it takes a little bit for cancer to develop, so we didn't want our prevalence values to be distorted out from all the malnourished pups that were coming through the center, or animals dying just because they were a failure to thrive. So we just looked at subadults, juveniles, and adults within the cancer aspect stuff. So Denise Greger actually published another paper that looked at the prevalence from 1991 to 2000, and that showed that 16% of the animals had urogenital carcinoma, and so the prevalence was pretty stable throughout that time period. The numbers, if you look at them, so 91 animals had cancer with Denise's paper, and 67 had them during Francis's time. If you look at the amount of animals that we saw from 2005 to 2015, we saw 215 animals within that 10-year time frame that had cancer. So if you're a vet or a biologist that's doing the necropsies on those animals, you think you're seeing cancer way more, but you need to look at the denominator. How many sea lions were actually coming through the center? So you need to look at the prevalence of it. So we actually saw in the past 10 years 932 animals, necropsied, 932 animals. So it's percentage-wise, although the prevalence has increased, it has not increased significantly. So it looks like the disease has been pretty stable for the past 40 years, 50 years. But we are getting more potential samples that are coming into the center because the sea lion population is growing, and our rehab efforts and rescue efforts are getting better. And this is just a breakdown, and one of the reasons why I really wanted to put this graph up is because it shows you pictorially how important that denominator is when you're trying to figure out prevalence. As you can see, the prevalence for the most part was anywhere between about 20 to 40%. But a couple years it dropped down to 11% or 16%. Does anyone know what those years potentially were or what they represent? Why we saw so many more animals those years? Those are El Nino years. So those El Nino years are what's causing the domoic acid outbreak. So we're getting floods of animals that are having seizures within those years. So if you just were to look at the prevalence numbers, you would say, oh, in 2009, you only had 11% prevalence of cancer. Maybe, you know, the cancer is starting to go away. No, we actually were just seeing more stranded animals within that year, and the cancer numbers kind of remained steady. So it's something that I've learned you can make statistics work for you or against you. So you really have to understand your data set as a whole and where your samples are coming from. This is a very biased sample population. We're just looking at the animals that are stranding on the beaches that are coming into our hospital. So you have to interpret the data with that in mind. But overall, the whole point is that urogenital carcinoma has remained pretty consistent within this population over those years. So here's a breakdown of the numbers from that paper. these are only animals that were necropsied at the center because, like I said, it's hard to see whether or not the animals have cancer pre-mortem. So we don't say they do or don't have cancer if they're released because we just don't know. So these are the sample size that we're going to be talking about today are just the animals that were necropsied. So of the 985 juvenile and subadult animals that we saw, 2.6% of them had urogenital carcinoma. Although that doesn't seem like a large percentage, for a child to develop cancer at a 2.6 rate, if 2.6% of the children within our hospitals had cancer, that would be an alarming rate. In humans, it's 0.00001% of children develop cancer. So it just goes to show that there's probably an infectious etiology that's driving this, and there's probably an immune suppressive thing that's happening within this population, the fact that there's so many young animals developing this cancer. Of the juveniles, it was split evenly between the males and females. You're just as likely to get cancer if you were a boy than if you were a girl. And in those cancer cases, 77% metastases were seen. And it was the cause of death in all but one of these cancers. So in the adults, it was at a 10-fold higher prevalence. So 23% of the 932 adult animals that we necropsied had urogenital carcinoma, which is obviously significantly higher than the juvenile and subadults. And metastatic disease was considered to be the primary cause of death pretty much in all of those animals. But that's just the nature of the beast. They're stranding in late-stage disease. I will say that we did see some early-stage disease, and that's very useful in studying this, and I'll talk about why in the next slide. And then overall, the breakdown of all of the animals put together, we necropsied within those 10 years almost 2,000 animals. And we saw a split between the males and females of about 1,000 females, 914 males. What's important to note, though, is we get, just based off of the natural history of the animals, way less males stranding along our coastline. So it was actually the adult males that had the highest prevalence of cancer, where 1 in 3 of the adult males that came through the Marine Mammal Center had urogenital carcinoma. And then of the females, about 1 in 4, 1 in 5 of the females had urogenital carcinoma. And then if you kind of combine the juveniles, the subadults, and the adults and get a prevalence of that, about 13% of the animals that came through the center had urogenital carcinoma in that 10-year period. So like I said, these domoic acid blooms are terrible because these animals show up seizing on our beaches, and a lot of times we cannot bring them out of it, but that also is really good for my research because then we can get samples from animals that are in early stage disease. And trying to understand the difference between what's going on in early stage cancer and what's going on after it's metastasized is really where the key is. So the Marine Mammal Center has done an incredible job throughout its history of prioritizing research. They're very well aware that although they're making a difference for one individual animal in rehab, they're actually not making a population level difference. It's a healthy population, so they're taking advantage of these animals showing up on the beaches to better understand what's going on out in the environment, and that's always their highest priority. So they do full histopath on almost all of the animals that come through there. All those 2,000 animals had histopath. And having the samples that are in early stage disease are really hard to catch. You can't even see the lesions in late stage disease sometimes. So by taking a sample of a reproductive tract and doing the histology on it, even though it looks normal, will tell you for sure if that animal had cancer or if it didn't. And if they weren't doing histopath regularly on those reproductive tracts, they would have missed about 20% of the cancers that came through the center. So that's my push for I wanted to be a pathologist. I'm not sure I'm smart enough to be, but they have all the answers, and pathology is so important. So that's my push for doing histopath even when you don't think something's wrong. But of the advanced stage disease, it made up about 80% of the animals that came through. And basically, if they had an invasive lesion, it had metastasized throughout their entire body. And these are kind of the sites where it metastasized. Like I said, it's the metastases that will kill you. So it's a pretty normal spread through the blood and the lymphatic system based off of the organs that you see here. So the lymph nodes are most commonly affected, particularly the sublumbar lymph nodes. So that's right underneath the backbone. It makes sense that it would go there first. And then it goes to the lung. It's actually so impressive to see some of these lung tissues. You have no idea how these animals are breathing, where it just is like one solid tumor. And these animals they come in poor body condition, but they're still out there living. So it's crazy they even have enough surface area for gas exchange. And then it kind of spreads throughout the body from there. Clinically speaking, like I said, it frustrates me because sometimes I just can't figure out that they have cancer. But of the animals with advanced stage disease, about 63% of them had the hydronephrosis. That's great because I can put an ultrasound probe on them and see that their kidneys are swollen with urine. And if they have hydronephrosis, it's pretty much a slam dunk that they have urogenital carcinoma. Makes me feel a lot better. I can sleep at night. I know I euthanized that animal for the right reason. They also come in with some pretty pathognomonic clinical signs, and although you have to rule out things like trauma, hind leg swelling, I think I actually have, the hind leg swelling, which is happening back here, that abnormal tail position where it's held up like that and they drag, they get paresis of the hind limbs. It's probably associated with pain and then lack of lymphatic drainage back up to their system because those lymph nodes get so enlarged in their tiny little pelvises that they get this pitting edema in their hind legs. But 50% of the animals with advanced stage disease presented with that, as well as the genital or vulvar swelling. And with the males, they can have penile prolapse. And then state of nutrition clinically, it's not really that relevant because although I like to think I'm really good at assigning a state of nutrition on an animal, I can't assign the difference between a 1.98 and a 2.35, but it does go to show that those animals are very cachexia, muscle-wasted and thin when they come in, and typically the thinner the animal is, the more likely that they have cancer. Oh, and that's Moon Unit. She's my poster animal. I have her growing in cell culture. She lives on. So the purpose of my PhD research, I'm finally getting to it, was to figure out if odorine herpes virus 1, which is the gamma herpes virus associated with this cancer, actually plays a role in urogenital carcinoma or not. So my primary goals for my PhD were to sequence this genome using the cool new molecular techniques that are now available. You would not have been able to do what I did the past 3 years, 10 years ago, within the time frame or with the budget that I did it in. The techniques that are coming up and available right now make it possible to do research in non-model organisms in ways that were never possible before. And identifying possible oncogenes as well as assessing the expression of those oncogenes within the tumor samples. So viruses, about 50 to 20% of cancers in humans and animals are associated with viruses. A lot of people don't realize that. And viruses are so cool because they basically hijack a cell. And they're responsible for cell cycling. The viruses can take over a cell's ability to cycle and replicate in order to make baby viruses. They can also prevent a cell from dying. So that's apoptosis, controlled cell death. Viruses can basically make cells proliferate and then make them live forever. And that's cellular transformation. Once you transform and you can live forever and you're cycling like crazy, then you can develop additional mutations that can develop into neoplastic transformation. So that's what viruses do. There's a variety of viruses that can be associated with cancers, but we're going to be focusing on the herpes virus. And a lot of people don't really understand what cancer is, but if you understand this list, this is how things develop into cancer. It's a self-sufficiency from growth signals, an insensitivity to inhibition growth signals. They can evade apoptosis, limitless or immortal replication, sustain angiogenesis, so that means they can attract a blood supply into the area because they're replicating so much, they need the nutrients in order to do so, so they recruit blood vessels to grow into them, and then the invasion and metastasis, which is key to the mortality of the animal. This picture is just to go to show you that there's a lot going on, and there's a lot of repetitive ways to control cell cycling, gene regulation, and apoptosis, and that's because you don't want to die if you're not supposed to, and you don't want to not die if you're supposed to. And viruses kind of can play with a variety of molecules within these different pathways in order to control cells, and oftentimes they do it in multiple areas redundantly to make sure the cell's going to live, the cell's going to evade the immune system, and that the cell's going to allow it to proliferate viruses. So herpes viruses, they're double-stranded, large genomes. They're about 120 to 170 base pairs, so nucleotides in length, when other viruses like papillomaviruses or morbilliviruses are only like 4,000 to 7,000 base pairs. So herpes viruses are huge. And because of that, they have a lot of different genes, and then they can do fancy things with those genes. And that's why they're associated with cancer. So there's 8 human herpes viruses. I can guarantee you everybody in this room has one. It depends on what flavor you have, depending on how it impacts your day-to-day life. 2 of these 8 human herpes viruses are associated with cancer, and that's Epstein-Barr and Kaposi sarcoma. Epstein-Barr causes lymphomas and nasopharyngeal tumors in people, and Kaposi sarcoma is associated with AIDS. So it's the tumors that AIDS patients often succumb to. And the point to me pointing out these 2 cancers these are the ones that we're comparing the sea lion cancer to understand what's going on with them. They have very specific genes that have been already worked out to be associated with the oncogenesis of those viruses. So herpes viruses are very species specific. They typically don't jump hosts. We will not catch the sea lion herpes virus, which I am very happy about. And they co-evolve with their host. And the reason why they're so species specific is because they can actually steal genes from the host genome and incorporate them into their own genome. And then that allows them to control the host cell cycling. And although we all have the same proteins that are responsible for controlling the cell cycling, they're slightly different in every species. So that's what makes these herpes viruses species specific, is because they've obtained genes from their host to control these cell cycles. And most herpes viruses are made of core genes, so there's a group of genes that make you a herpes virus, and then there's the accessory genes, and those accessory genes are what make you different from other herpes viruses. So it's the accessory genes that we're interested in here. So I'm definitely not going to get into details of my research because it's too much now, but we did next generation sequencing. We used a technology called MiSeq and PacBio sequencing, and that allowed us to sequence the whole genome of the virus. And we did something called a de novo assembly. My PhD was very heavily focused in bioinformatics, and I spent about a year of my life putting together this little gene map of that herpes virus. So although it may not be very impressive to you, it is like on my refrigerator, and I'm very proud of it. So the genes that are in red are the core genes, and the genes that are in yellow and blue are the accessory genes. And the cool stuff, sometimes herpes viruses can integrate into the host genome, and then when they pull themselves out, they can pick up host genes on the end of that genome. So that's why the cool genes kind of live on the ends of the genomes, and that's the genes that we're going to talk about in a little bit of detail. This is just as a map to show you, when you look at the core genes, and that's to understand the phylogeny of how these herpes viruses are related to each other, so you want to look at the genes that are more conserved within the herpes virus families, you can see that down here, there's dolphinid gamma herpes virus 1, which is associated with genital lesions in dolphins, and then OTHV1 here, which is the one that I do research on. It's kind of like an ocean clade. So it looks like this herpes virus did not exist in evolutionary time frames when this, or didn't cause cancer, or was different than all of these herpes viruses when the host evolved to go into the water. And then it broke off and started causing issues for some of these animals. I could explain that in more detail for people that are into phylogeny, but I'll spare the rest of you. So there was a variety of interesting potential oncogenes that we wanted to look at, that we identified as possible oncogenic genes. So I'm not going to go into details of any of them, but here's a list of them, just so you know some acronyms that I'll be talking about briefly. So my main question was, is this virus actually inducing the cancer, and how do we determine if it is? So we use this new technology called RNAscope and BaseScope. This is the coolest thing in the world. Immunohistochemistry is something that's really important. It's a way that we can tag specific molecules that we're interested in looking at. Typically, you use antibodies for that. And in order to develop those antibodies, you have to develop it within a lab. You have to infect a mouse with an antigen. You have to pull out those antibodies. It's a labor-intensive, time-intensive, cost-intensive process. This technology, you can actually design nucleotide probes to specifically bind to your protein or RNA of interest. And they can basically, as long as you have the gene that you're interested in, you can design these probes and they can get them to you within 2 weeks. It's a little bit expensive, but in the big picture of things, it is not expensive at all in comparison to developing antibodies. Base scope is a little more sensitive than RNA scope, and it's a little newer technology. Because we are looking at genes that the virus has stolen from the host, we needed to make sure we are looking very specifically at the virus genes, which have evolved slightly from what the host genes look like. So the base scope allowed us to refine what we are looking at a little bit better. And now I'm going to show you guys some cool pictures. So this is a normal H&E slide of a cervix of a sea lion. This is healthy. The cells are doing what they're doing. Although it looks like it's invading, those are actually the folds of the cervix going in. So they're not going past the basement membrane. They're doing what the cells should be doing. So basically, we took these probes. We designed some probes to look at some housekeeping genes, which were our positive controls, the genes that should be normally regulated within the cells, and then looking at our oncogenes. So in this healthy animal, this is a housekeeping gene, so a gene that we're just expecting to be expressed. This is a gene associated with DNA proliferation. And then this was our oncogene probe. So I should mention that these little pink dots are the positive stains, if you guys can see them up there. And when we stained it with our oncogene probes, all the healthy animals came out negative. So none of the healthy animals had these herpes virus oncogenes associated with them. This is like the poster child of my PhD. This is normal epithelium, and this is it just developing into cancer. So you can see this uncontrolled cell growth. It's diving past the basement membrane. There's a bunch of inflammatory cells here. The immune system's not happy with how this tissue is acting. So healthy on the left, diseased on the right. This is the control probe, so you can see, it's not the same exact picture, but over here there's a little bit of healthy tissue. You can see the probes picking up, but it's pretty homogeneous throughout. This is the oncogenes. So basically there's no pickup of this herpes virus oncogene within the healthy tissue. However, throughout the cancer cells, this oncogene, which is a cyclin-dependent kinase 4, is highly upregulated. Like, this is really, really highly upregulated staining within just the cancer tissue, which is kind of a slam dunk that tells you, yes, this herpes virus is inducing this cancer. And then this is just a carcinoma in situ, so this is not invaded past the basement membrane, but is proliferating faster than it should be. The positive control kind of homogeneously throughout the epithelium. And then this is a V-flip oncogene, which is a very important oncogene associated with human cancers, and it's only upregulated within the cancer areas of the diseased animal's carcinoma. And then this is an invasive lesion. This is just all tumor, basically, with a little bit of stroma throughout on the H&E. And this is the positive controlled probe, and that's the oncogene. So although I have to do a lot of analysis and statistics to prove that, it's upregulated, it's pretty easy to see just looking at these results that, yes, the oncogenes are clearly upregulated within the cancer and not within the healthy. So basically, my question for my PhD is OTHV1 associated with urogenital carcinoma? And the answer is yes, absolutely. I will say that it is not the only thing, though. There are immune system components and suppression that are allowing this herpes virus to grow out of control. So although my PhD was very focused on this herpes virus, there's a ton of other components that are at play, and it's not just this herpes virus that's inducing cancer. And it is a good comparative model for studying virally induced diseases, particularly metastases. And the fact that these animals have this intact immune system, have a representative microenvironment, it allows us to understand how all of these cofactors can be at play in the association of the development of the disease. So if we can figure out ways of treating these sea lions to block the metastases, or better understand how the virus is changing a normal cell into a cell that has uncontrolled cell growth, you can aim your future treatments at those pathways, and that's something that goes across the board in cancer. Those cell proliferation pathways are very highly conserved across species. And then my conclusion is this, like I said in the beginning, this isn't just sea lions where you can do stuff like this with. Aquatic animals are excellent sentinels for environmental health. And the reason why these sea lions are getting cancer is not just because they have these herpes viruses. It's because they're immune suppressed as a result of their environmental exposures. So urogenital carcinoma is a great model for understanding what's going on with the cancer in them. But the malnutrition in the pups and the domoic acid that we see, the increased outbreaks that we're seeing, are all representing a bigger issue issue that is going on out in the environment. And so it's a much easier way to explain to the general public why we need to be protecting the environment, because those environmental health issues are impacting not only the animals that live within that environment, but people as well. And other marine mammals besides sea lions are something that you can look at this with. By doing appropriate stranding response, rehab, and necropsy of animals that show up on your beaches, those are pretty much free lab animals that have just shown up. You should never let those samples go to waste. Doing health assessments on healthy populations as well as impacted populations are super important. Understanding a baseline of what a system should look like before it's actually had a negative impact is great, but studying things that have been negatively impacted and trying to figure out how they're getting out of that is also really important. And I know I focused a lot on marine mammals, but the fish and the invert stuff is actually probably more important to look at than the marine mammals, because you're understanding the biomagnification of things and how the food source is having an impact on those animals. So those are all of my funders and the people I collaborate with, and I will take any questions you guys have. I know that was a lot, I'm sorry.
Curious about the thing about DDT,
Mm-hmm.
and it was banned so long ago, so I'm wondering where it's coming from. It's coming from some of the southern countries of Mexico, et cetera, or any other country that still has DDT in there. This old,
Yeah.
old DDT.
It sticks around in the environment, unfortunately, for a really long time. It is still used in Mexico, and that definitely makes its way up. And PCBs in particular in that area, there was a lot of bombing practice and stuff, like in the 40s to 70s within the California Bight. So between pesticides for the DDT and the bombing within that area, the sediment within that area is just laden with these contaminants. So it just bioaccumulates, and it's going to be an issue, a long-term issue. Like the DDTs won't even start going away for 50 to 200 years. So although we've stopped using it, that's great, but it's still impacting animals significantly, and there's still, we don't use it in the US, but it's used all over the world, unfortunately. Anyone else?